Walking is among the most consistently recommended health behaviors in medicine and among the most consistently underestimated. The recommendation is so familiar, delivered so routinely, that most people have stopped hearing it as meaningful information. It sounds like the advice you give someone when you have nothing more specific to offer: gentle, low stakes, optional. The biology tells a different story. A daily walking habit does not simply burn calories. It produces measurable structural changes in the brain. It directly improves how skeletal muscle handles glucose. It reshapes the hormonal environment that determines how the body responds to stress. It stimulates mitochondrial biogenesis, the production of new cellular energy machinery. It maintains the joint, balance, and neuromuscular systems that determine how long a person remains physically independent. These are not incidental benefits of light activity. They are specific, documented biological adaptations to a specific type of movement stimulus. What follows is an explanation of what the research shows happens inside the body when walking becomes a consistent daily habit. Not motivation, not general encouragement, but the specific mechanisms behind each effect, and why consistency is the variable that determines whether any of them actually occur. The most striking evidence for walking's health impact concerns the brain, and specifically a structure called the hippocampus. The hippocampus is a bilateral structure in the medial temporal lobe involved in the formation of new memories, spatial navigation, and learning. It is one of the first brain regions to show atrophy in age-related cognitive decline and in Alzheimer's disease. For most of the 20th century, the prevailing assumption in neuroscience was that the adult brain could not generate new neurons, that the loss of hippocampal volume with age was irreversible. That assumption has been substantially revised. A landmark study published in the Proceedings of the National Academy of Sciences randomized older adults to either a walking intervention, moderate-paced walking for approximately 40 minutes 3 times per week, or a stretching control condition. After 1 year, participants in the walking group showed a measurable increase in hippocampal volume on MRI. The stretching group showed the expected age-related decrease. Walking had not merely slowed the rate of hippocampal atrophy, it had reversed it. The mechanism behind this finding centers on a protein called brain-derived neurotrophic factor, or BDNF. BDNF supports the survival, growth, and maintenance of neurons and promotes the formation of new synaptic connections. It is sometimes described as a neurological growth factor, a compound the brain uses to build and maintain its own infrastructure. Aerobic exercise, including walking, consistently elevates BDNF levels, and the increase in hippocampal volume observed in walking studies correlates with the elevation in BDNF. The practical consequences are directly observable. People who walk regularly report improvements in memory clarity, mental focus, and the ease with which new information is retained. These are not subjective impressions produced by feeling healthier. They are reflections of measurable changes in the organ producing them. Stress is not only psychological, it has a specific hormonal architecture, and that architecture has physical consequences. When the brain perceives a threat, real or anticipated, the hypothalamic pituitary adrenal axis activates and triggers the release of cortisol from the adrenal glands. Cortisol is appropriate and functional in acute situations. It mobilizes energy, sharpens attention, and prepares the body to respond. The problem is chronic elevation. When cortisol remains persistently high, as it does in people living with sustained psychological stress, it produces a cascade of damaging downstream effects: disrupted sleep architecture, suppressed immune function, increased abdominal fat deposition driven by cortisol's gluconeogenic activity, and progressive impairment of the hippocampal tissue discussed above, since cortisol at chronic high levels is directly neurotoxic to the hippocampus. Walking interrupts this cycle through a specific mechanism. The rhythmic bilateral repetitive movement of walking activates the parasympathetic nervous system, the division of the autonomic nervous system responsible for rest, repair, and recovery. The parasympathetic system is the physiological counterpart to the sympathetic fight-or-flight response. When it is activated, heart rate slows, blood pressure decreases, cortisol production is suppressed, and the body transitions from a state of alert mobilization to a state of regulated recovery. The rhythmicity of walking is specifically relevant to this effect. The predictable, repeating left-right movement produces a pattern of sensory input that the nervous system registers as compatible with safety. Walking outdoors amplifies this further. Natural light exposure, distance vision, and varied sensory environment all contribute additional signals that reinforce parasympathetic activation. Walking does not resolve the sources of stress. It changes the body's physiological relationship to them. A person who walks regularly does not eliminate stressors from their life, but their nervous system is demonstrably less likely to remain in a state of sustained activation in response to those stressors. A well-known study from Stanford University tested creative output in individuals who were sitting versus walking using standardized measures of divergent thinking, the capacity to generate multiple solutions to open-ended problems. Walking increased creative output by approximately 60% compared to sitting. The effect persisted briefly even after participants sat down following a walk. The mechanism involves two converging factors. First, walking increases cerebral blood flow by engaging the calf muscle pump, the contraction and relaxation of the large muscles of the lower leg that act as an auxiliary pump for venous return, sending blood back toward the heart and increasing cardiac output available to the brain. More blood to the brain means more oxygen and more glucose available to the neural tissue performing cognitive work. Second, walking activates what neuroscientists call the default mode network, a distributed set of brain regions that become most active when the mind is not focused on an external task. The default mode network is associated with insight, mental simulation, making novel connections between disparate ideas, and the kind of associative thinking that produces creative solutions. Forced, effortful concentration tends to suppress this network. Walking, which requires enough motor attention to keep the mind occupied but not enough to demand sustained executive focus, allows the default mode network to operate freely. This is the neurological basis for the common experience of solving a problem during a walk that resisted solution while sitting at a desk. The answer was not produced by thinking harder. It was produced by giving the brain the conditions under which associative thinking operates most naturally. Every meal that contains carbohydrates produces a rise in blood glucose. Insulin, released by the pancreas in response to that rise, facilitates the uptake of glucose by cells throughout the body, primarily by muscle tissue, which is the body's largest glucose sink. Over time, when muscle tissue is chronically underused and dietary glucose is chronically elevated, the cellular machinery that responds to insulin becomes less sensitive, a condition called insulin resistance. Glucose remains in the bloodstream longer after meals. The pancreas compensates by producing more insulin. The cycle reinforces itself. Walking directly addresses this through a mechanism that operates independently of insulin. When skeletal muscle contracts during walking, it activates specific molecular pathways, particularly AMPK, adenosine monophosphate activated protein kinase, that facilitate glucose uptake into muscle cells without requiring insulin signaling. The contracting muscle essentially pulls glucose out of the bloodstream to fuel its own activity through a pathway that bypasses the compromised insulin response. Research has demonstrated that walking for as little as 10 minutes after a meal can significantly reduce post-meal blood glucose elevation, in some studies more effectively than a single longer bout of exercise performed hours after eating, because the timing of the walk coincides with the peak of the glucose absorption curve. The muscle is contracting and demanding glucose at precisely the moment that glucose is entering the bloodstream from the gut. The signs of impaired glucose handling are often misattributed to other causes. Afternoon energy crashes, persistent sugar cravings, reliance on caffeine to sustain alertness, drowsiness in the hour following a meal. These are not personality traits or signs of poor discipline. They are metabolic signals. Walking after meals consistently directly addresses the underlying mechanism producing them. Energy production at the cellular level depends on organelles called mitochondria, the structures within each cell that convert oxygen and nutrients into ATP, the molecule the body uses as its primary energy currency. Mitochondrial function declines with age and with physical inactivity. When mitochondria become damaged or dysfunctional, the efficiency of ATP production decreases. Fatigue results not from inadequate rest, but from inadequate energy production. The cells are receiving fuel but cannot convert it effectively. Walking stimulates two distinct mitochondrial adaptations. It promotes mitochondrial biogenesis, the production of new mitochondria, through activation of a transcriptional coactivator called PGC1A, which is upregulated by sustained low to moderate intensity aerobic exercise. And it stimulates mitophagy, the selective removal of damaged, dysfunctional mitochondria, allowing the cell to replace aging energy machinery with new, more efficient structures. The quality of energy produced by this process is distinct from the energy produced by stimulants. Caffeine and high-sugar foods produce energy by borrowing against the body's hormonal and metabolic reserves, spiking cortisol and blood glucose to create temporary alertness that is followed by a crash. Mitochondrial efficiency produces a different subjective experience, calm, sustained capability that does not depend on external inputs and does not produce a withdrawal state when those inputs are absent. People who establish consistent walking habits frequently report this shift in energy quality, often describing it as feeling capable rather than wired. The digestive system relies on peristalsis, rhythmic muscular contractions of the intestinal wall that propel food through the gastrointestinal tract. These contractions are stimulated by movement and suppressed by prolonged inactivity. Sitting for extended periods after meals slows gastric emptying and reduces intestinal motility, contributing to bloating, discomfort, and constipation. Walking stimulates peristalsis through two mechanisms: the mechanical effect of abdominal muscle engagement with each stride, which applies gentle rhythmic pressure to the digestive organs, and the neural effect of parasympathetic activation, which enhances digestive function. The parasympathetic system is sometimes called the rest and digest system precisely because it governs the biological state in which digestion operates optimally. The recommendation to walk after meals, present across multiple traditional health systems and now supported by the glucose research described above, reflects a physiological reality that has been empirically validated. Movement after eating produces measurably better metabolic and digestive outcomes than inactivity after eating. The cardiovascular benefits of walking have been directly compared to those of running in large population studies. When total energy expenditure is matched between walking and running cohorts, the two activities produce comparable reductions in cardiovascular risk, including similar effects on blood pressure, LDL cholesterol, HDL cholesterol, and incidence of coronary artery disease. Walking achieves these benefits at lower mechanical stress on joints, lower cortisol elevation, and a much higher level of long-term sustainability than running. The mechanism through which walking improves cardiovascular markers involves both mechanical and molecular pathways. The increase in blood flow produced by walking creates shear stress on the walls of blood vessels, a mechanical stimulus that prompts the endothelial cells lining those vessels to upregulate nitric oxide production. Nitric oxide relaxes vascular smooth muscle, reducing arterial stiffness and blood pressure. Regular walking essentially provides the vascular endothelium with a consistent training stimulus that maintains its capacity to respond to flow demand, preserving arterial flexibility that would otherwise decline with age and inactivity. Consistency matters more to these adaptations than intensity. A 20-minute walk performed 5 days per week provides a more consistent and sustainable stimulus to the vascular endothelium than an intense workout performed once, followed by 4 days of inactivity. The walking surface of joints, the cartilage that covers the articulating surfaces of the knee, hip, and ankle, has no direct blood supply. It receives oxygen and nutrients through a process called imbibition. The mechanical compression and release of the cartilage during movement drives synovial fluid in and out of the tissue, delivering nutrients and removing metabolic waste. Movement is the mechanism through which cartilage is fed. Prolonged inactivity, therefore, does not protect joints. It deprives them of the nutrient delivery that depends on movement. The stiffness that accumulates after extended sitting is in part a reflection of this nutritional deficit in the cartilage. Walking, by providing regular cycles of joint loading and unloading, maintains the imbibition process and supports cartilage health. Walking also maintains the muscular support around joints. The muscles crossing the knee, particularly the quadriceps, protect the joint from impact forces and maintain its alignment during movement. Weakness in these muscles is a primary risk factor for knee pain and joint deterioration. Walking maintains the strength and coordination of these muscles at a level of stimulus that is sustainable and joint compatible in a way that more intense loading is not for many older adults. Walking produces consistent and measurable changes in the neurochemical environment that determines mood. It increases circulating levels of serotonin, which regulates mood stability and emotional resilience. It stimulates endorphin release, though the endorphin effect is more modest at walking intensity than at higher exercise intensities. It reduces cortisol and the inflammatory cytokines, TNF-alpha, IL-6, IL-1, that are directly associated with depression. And it elevates BDNF, the neurological growth factor that supports hippocampal function, which is consistently reduced in people with major depressive disorder. Multiple large systematic reviews and meta-analyses have found walking to be an effective intervention for mild to moderate depression and anxiety, not as a motivational tool or a behavioral distraction, but as a direct modifier of the brain chemistry that determines mood. The effects are comparable in some studies to those produced by antidepressant medication in mild to moderate presentations, without the side effects associated with pharmacological treatment. The mechanism is not distraction from negative emotions. It is a change in the biological conditions that shape emotional experience. A person who walks regularly is operating from a different neurochemical baseline than a person who does not. The most underappreciated dimension of walking's health value is its relationship to health span. Not how long a person lives, but how long they remain physically and cognitively independent. The decline that most people associate with aging is not typically a single catastrophic failure. It is a progressive disconnection. Muscles weaken, balance deteriorates, reaction time slows, confidence in movement decreases further, and the decline accelerates in a self-reinforcing cycle. Falls become more likely. Recovery from illness slows. The capacity to perform basic activities of daily living without assistance narrows. Walking interrupts this cycle by simultaneously maintaining multiple systems that age in parallel. Leg strength, particularly in the muscles protecting the hip and knee, is preserved by the regular loading of walking. Balance is continuously challenged and maintained by the proprioceptive demands of navigating varied terrain. Bone mineral density is supported by the weight-bearing nature of walking, directly opposing the progression of osteopenia and osteoporosis. Reaction time and neuromuscular coordination are maintained by the continuous coordination demands that walking places on the nervous system. Long-term epidemiological studies consistently show that people who walk regularly maintain functional independence for longer than those who do not, getting out of chairs more easily, climbing stairs without assistance, recovering from illness more rapidly, and sustaining cognitive function into advanced age. The effect is not attributable to any single mechanism, but to the simultaneous maintenance of multiple systems by a single, sustainable, daily activity. The final and perhaps most important point is that the biological adaptations produced by walking are not produced by occasional or irregular walking. They are produced by consistency. The body's adaptive systems respond to repeated signals, not to isolated events. When a behavior is occasional, the body treats it as noise and produces no lasting adaptation. When a behavior is regular, the body treats it as a defining feature of the organism's environment and adapts accordingly. Insulin sensitivity improves because the muscles are regularly being called on to manage glucose. Mitochondrial capacity improves because the cells are regularly being asked to produce aerobic energy. Cortisol regulation improves because the nervous system is regularly receiving the parasympathetic signal that walking produces. BDNF elevation and hippocampal maintenance occur because the brain is regularly receiving the blood flow and growth factor stimulus that walking provides. A 15-minute walk performed 5 days per week produces greater cumulative biological adaptation than a single 75-minute walk performed once, because the signal is delivered repeatedly across the week. A short walk after dinner performed consistently reshapes glucose metabolism more effectively than sporadic longer exercise, because the timing and frequency of the signal align with the mechanism it is targeting. This is why walking, despite its simplicity, despite the absence of equipment, despite the apparent modesty of the exertion required, is foundational rather than supplementary. It is the movement pattern the human body's biological systems were built to receive and respond to. It is sustainable enough to be performed daily for decades, and it simultaneously supports the brain, the cardiovascular system, the metabolic system, the musculoskeletal system, and the neurochemical environment that determines mood and emotional resilience, all from a single habit that requires nothing more than showing up most days and moving. The evidence does not describe walking as what you do when you cannot do something more demanding. It describes walking as one of the most broadly effective health interventions available, one whose benefits compound over years of consistency in ways that no other single behavior matches.